Welcome to Decoding the Data Ecosystem, a podcast with valuable insights, inspiring stories, and practical advice to guide you on your journey through the world of omics research and how the NIH Common Fund data ecosystem can support that journey. I'm your host, Dr. Alyssa Dillman, and I'm the Training and Engagement Director of the CFDE Training Center. My background's in bioinformatics, and I've been working in bioinformatics and data science training and engagement space for almost the past 15 years. I'm really passionate about lowering the barriers of entry for these fields through creative engagement activities such as these podcasts. Now let's get to the show. All right, hello. I'm here today with Malin Lindholm, who is currently serving as Junior Faculty and Director of the Human Molecular Moonshot at Stanford University, and Dan Katz, who is an Assistant Professor of Medicine in the Stanford Center for Biomedical Informatics Research and the Cardiovascular Medicine Divisions. So hello to both of you, and thank you so much, and welcome to our podcast. I would love to have each of you tell a little bit more about yourselves and your work. Let's start with Malin first.
Yeah, hi. It's great to be here and nice to meet you. My name is Malin Lindholm. I've been working on molecular exercise physiology research for almost 20 years now. Started with my PhD in physiology and medicine at the Karolinska Institute in Stockholm, Sweden, and then continued to do a postdoc at the Cardiovascular Institute at Stanford. And that was the time when this large initiative that we're going to talk about today, MoTrPAC, started. So I've been working on that for many years now, and transitioned to junior faculty, and now I'm a senior research engineer and director for what we call the Human Molecular Athlete Moonshot, which is part of the WOTSAI Human Performance Alliance. So I've always been fascinated by the immense impact of exercise on human health, and how something as simple as physical activity can lead to such immense health benefits and affect basically the entire body. So that's really how I got into exercise research initially, and have continued ever since.
Awesome. Well, thank you. And again, really excited to have you. And also, as a fellow Karolinska alumni, it's always fun to talk to other people who also got their PhDs there.
Yeah.
Dan, tell me a little bit more about what you do.
Well, now I'm jealous that I didn't go to the Karolinska. It sounds like it would have been great. Well, thank you so much for having me. I really appreciate the opportunity to talk about this incredible project. I am much newer to exercise physiology and the exercise world relative to Malin. So I have taken a medical route. I went to medical school in Chicago, and then did internal medicine residency training and cardiology fellowship training in Boston. And then finally moved out to Stanford to do the last phase of my medical training, which is in advanced heart failure and transplant cardiology. So that's sort of my medical world. And in terms of my research, I really became interested in using high dimensional data to better understand heart failure. Heart failure is it's a fascinating syndrome and disease, but it's very heterogeneous. And so became interested early on in understanding it using high dimensional data to really understand subgroups within heart failure. And have gradually became more and more interested in using ever greater data sources, so multi-omic data, genetic data, proteomic, metabolomic data, to better understand heart failure. And when I got to the last phase of my training, that's when I learned about MoTrPAC, and because I was already at Stanford, which is the Bioinformatics Center for that big project, which I know we're going to talk more about. It was an opportunity to study exercise, which is what I think of as sort of the opposite of heart failure. All my heart failure patients, what can't they do? They can't exercise. So the thought was, well, boy, if you could figure out what's so great about exercise, maybe you could then help them do what they need to do on a daily basis. Because for them, putting away groceries, just getting to the car and from the car, that's exercise for them. And so if we can optimize that, maybe their lives can be a little better. So that's kind of how I got to it a little bit differently. And so now I am one of the co-investigators for the Bioinformatics Center for MoTrPAC. I do also do research on other other big cohorts to the TOPMed cohort as well, which is a big consortium focused on cardiovascular health cohorts that have sort of come together for whole genome sequencing. And of course, I see patients. I see heart failure patients, and I take care of patients with transplants and LVADs. So, yeah.
Awesome. Well, thank you as well for being here today. And hopefully, it sounds like you guys had very different backgrounds. So I'm excited to kind of hear sort of both sides of how those fit into the MoTrPAC program. So for the listeners who may not be familiar, I was hoping that you could provide sort of a high-level overview of what MoTrPAC is and what makes it such a unique initiative compared to maybe other exercise physiology studies that are out there. Ellen, do you want to go first?
Give a stab?
Yeah.
So MoTrPAC, it stands for the Molecular Transducers of Physical Activity Consortium. So it's an NIH Common Fund initiative. It started almost 9 years ago, so it's been going on for quite a while. It's a US-wide consortium, so it includes many different sites across the US, different universities that are focused on different aspects of the study. So from recruiting and testing humans, so human volunteers who are interested in participating in these exercise interventions that we do, sites that conduct animal studies. So for MoTrPAC, we're doing a rat study to be able to look, for example, at organs that we can't access in humans. There is also sites that are focused on different aspects of analyses, so assaying these different biosamples that we collect from the humans and the rats that are sort of have expertise in analyzing these different molecular assays. Sites for analyses, and we, as both me and Dan, represent the Bioinformatics Center, so we're really tasked with getting all of these data and building analysis pipelines to process those data in a consistent manner. And then we work with the entire consortium to analyze those data and then also share these data with the scientific community and also the rest of the, anyone who might be interested in this data, because data sharing is also an important aspect of what we do. So there's a lot of study coordinations. There are also study coordination sites and biorepositories that handle all of these samples, distribute to the sites that are going to analyze. And it's also possible for scientists out there. So if there are scientists listening to this, you can, if you're interested in MoTrPAC data and actually running assays on MoTrPAC samples, it is possible to contact the consortium and apply to be able to use those samples. So we step back and take like a 10,000 foot view. MoTrPAC really consists of these two main arms, the animals and the humans. These include the human part. I will get more into that. Dan has been focusing a lot on these analyses. They include both sedentary people that do a training intervention, either resistance or endurance type training for 12 weeks. They also do an acute bout of exercise before and after those 12 weeks. So we can look at sort of what are the transient changes that occur in association to one bout of exercise, as well as these longer term training induced changes. And we do it's a similar approach in the rats. The rats only do endurance training, but there we also look at both training effects over time and then what happens in response to one bout of exercise. What's nice with the animals, as I mentioned before, we can look at a lot of different organs that we can't look at in humans. And we can also get a better time resolution because we can get samples more frequently than we can for humans. So for the animals, we're looking at 19 different tissues, so a ton of tissues. And in humans, we're looking at muscle, fat, and blood, and look at lots of different levels of regulation, and what to really identify what are some of these key molecular transducers of the exercise response, all the health benefits that we see with exercise, and also start to look at what's the variability in this responsiveness, and can we start to try to predict who will respond better to what type of training. And I think we'll get into more of different aspects of MoTrPAC as we during the podcast.
Yeah, and I think the project is so tremendously large. I mean, if you step back and think about it, it's a project that has a randomized control trial, a full randomized control trial within it. And yet that isn't even probably quite 50% of the whole project, because as Milan alluded to, it's got the experiments around the rats that are doing, both endurance and are doing training and acute exercise. So the humans are doing a randomized controlled trial of exercise of two types, actually. So they're doing both resistance and endurance exercise. And they're also then randomized. There's a randomized group that is controlled, that sits and does nothing, so that we can actually see what are the true exercise effects. So they're the ones that get to sit and read a book, while the rest of the group is doing what I am told is some pretty challenging exercise. This is not light exercise. The humans that are going through the resistance arm are doing, they're doing 8 exercises. They're doing 8 to 12 reps, 3 sets on each one of those, but they need to stick to a time regimen. So they're getting quite out of breath, actually, as they're doing this exercise. A lot of people in the consortium actually did the resistance exercise, and they got out of it like, ooh, that was hard.
I think I'd sign up for the read a book group. That sounds great.
Yeah. Well, the beauty of a randomized controlled trial is that you don't get to sign up. You have to actually do with the one we assign you to. As much as people were hopeful, actually, they were hopeful to be in the exercise arm. Some were a bit disappointed when they got randomized to control. But so there's this randomized control trial.
Did some people use this as sort of like a motivation to get more exercise? Like, was there a question on like why did that come up out of curiosity?
Yeah, I think it was definitely a big motivating factor. There were a lot of people, like I said, when they found out that they were randomized to the control group, they were a little bit disappointed, because they were saying, I'm going to get 12 weeks of really dedicated personal training, either in endurance or resistance exercise, which is something that they felt like they needed. You will see in the data that safe, fortunately didn't lead to sort of sampling bias, because I mean, you imagine people don't get randomized to the group that they want, that they just drop out. We don't think that has affected the data, fortunately. But I think ultimately, they did an exceptional job of keeping these people engaged and working in what is a really rigorous exercise bout. And then, outside of just this randomized control group in the adults, which are really focused on sedentary adults, people who are not active, there's 2 additional arms that were the pediatric cohort. So there's adolescents from age kids really 10, and then adolescents as well, 14 to 17. And then they are divided up into people that were children that were a little bit more low active versus a little bit more high active in terms of what they were doing in their lives. And then the adults, similar, there's another cohort, non-randomized, but another comparator cohort of highly active adults who basically signed up and said, yeah, endurance exercise, I do regular highly active exercise. And so regular resistance exercise, excuse me. And they serve as a comparator to say, if you go through 12 weeks of training as a sedentary adult, how close do you look to the highly actives? Do you look like them, or does 12 weeks get you 50% of the way, 100% of the way, or does it look completely different? That's a big question that we don't know the answer to, that MoTrPAC will help us answer.
So I have sort of a silly follow-up question. So obviously, when you first asked them about their general lifestyle habits, what happens if the folks that got sort of like randomly assigned to the non-activity group, like what if they're like slowly beeping up their activity like outside of your experimental time? Like how do you sort of like query them throughout to see if they've like sort of like bumped up their own like personal exercise time?
Yeah, so we do watch that pretty closely. So they actually wear actigraph accelerometers. So during the exercise, throughout and at various points throughout the study, their activity is sort of monitored so that we can track a little bit of that. There's check-in calls. There's a chance to check in, but that's actually part of one of the values of the control group is that we do need to kind of control for people just being under watch, this is a known effect, right? In all trials, as soon as you enroll in a trial, you start to change your behavior a little bit. So we actually have, that's one of the many benefits of the control group in the randomized part of the trial, that they can kind of help us control for some of those effects. But we do try to keep them as steady as him. And we actually also ask them not to lose weight. That's not part of the study. right? We don't want them to change their diet, to lose weight. We're trying to focus entirely on the exercise effects.
Exercise. One other sort of basic question about this data set. Why rats as opposed to other model organisms?
Yeah, that's a great question. And the most common animal that we use is mice. And the problem here was that, mice are much smaller than rats. And one of the unique aspects of MoTrPAC is to be able to look at all of these different levels of molecular regulation. So from the transcriptome to the proteome to the post-translational modifications, the metabolome, and also the gene regulatory aspects like the epigenome. And in the humans, we also look at the genome. But to be able to look at all of these different levels of regulation, we need a lot of material. So biomaterial. So that's one of the reasons for going with the bigger rats. And also this particular model, we used a cohort that was handled by the NIA, so the National Institute of Aging, that was able to provide all of the animals needed in a timely manner for the animal sites to be able to conduct these studies. And there was also experience from the people who were doing the animal intervention studies that have seen that the response to an endurance intervention in these rats. So what they've done is they've done treadmill running 5 days a week for the endurance training part. And the changes in aerobic fitness are at the similar level that we get with 12 weeks of training in the humans. So if you look at the percent increase in VO2 max, they do respond similarly. So that's why it was selected as a good model in this case. And of course, you can do bigger animals, but then becomes much more expensive and much more complex. And there are, of course, ethical considerations for all animal studies. So rats were sort of a good middle ground here.
Yeah, that makes sense. Okay, so now that we've got kind of like a big picture of sort of the, overall goal of MoTrPAC, I did want to back up a little bit because Marlene, you'd mentioned, there's lots of different tissues and collection techniques and, multi-time point sampling. How do you even manage all of that different kind of data, right? You got human and rats and multi-omics and multi-time point, and they're coming from all these different centers. Like, how do you balance all of that?
Yeah, that's actually one of the major challenges with MoTrPAC, and one that we from the Bioinformatics Center struggle with every day. It's I think the simple answer is you just start by taking it one step at a time, like one technique, one tissue. What are the sort of major effect we see for this particular assay and this particular tissue and this particular time point, and of course also the species aspect. Um, where do we see the most effect? Where do we see the least effects? Are there time points that are more affected, depending on, molecular assay or level of regulation? And then once we have taken all those steps, then we start to work with these more integrated approaches where we try to integrate these different multi-omic analyses to see if we can pick out, can we identify key regulators in here? Can we find things that are common between rats and humans, for example? And so far with MoTrPAC, we have mostly done analyses on the rat data and the endurance training response in the rats. We also have this acute exercise bout in the rats that we're analyzing now. The human data where the intervention study is done, and we have data from the first the people that were recruited prior to the COVID shutdown, looking at the acute bout of exercise. So we're sort of starting to get to that also interspecies comparison, but we're not quite there yet. What we have done with the rat data that's been published is, of course, also to compare to some other data sets that are out there. I mean, we try to utilize all the data that's been generated so far. I mean, there's a lot that's already been done in exercise science, and we're really aiming for MoTrPAC to build on that and provide new aspects.
Yeah, and if I can also just for a moment sort of take off the biology hat and put on the informatics hat a little bit. I mean, this is it's a real informatics challenge to put this together, and I'll sort of sing the praises of the Bioinformatics Center a little bit, which is that it requires standardization and really clear communication with all of the data sources. There's a lot of different, as Malen mentioned, with all the different omic types that we have, all the tissues, there's multiple sites that are generating very different kinds of data. Some are doing genetics, some are doing transcriptomics, metabolomics, proteomics, phosphoproteomics, proteomics of different types on different assays. So each one of them requires sort of a different adapter to get their data into the data ecosystem that we're building sort of just locally, much less the Common Fund. So it's important to have those standards set out early, and this is before I arrived. The Bioinformatics Center did a lot to create standards and rules and think ahead to how those data were going to all sort of play nice together. And so then once you have that, it makes it a lot easier to do some of the analyses that Malen was alluding to, because the data all takes on a similar format. And so that is part of having really fair data is ensuring that it's interoperable. That's the I in fair, right? Is that it's interoperable. And so ensuring that it has this sort of standard structure, exactly as Malen said, is something we think about every single day at the Bioinformatics Center, so that we can do these really high level analyses, like compare across species or across tissues in each set.
And I think I just wanted to also emphasize that this is a massive team effort, and it requires a lot of different expertise. And MoTrPAC is a big consortium, so we have a lot of amazing scientists with different expertise that are helping to put all of this together. So it's definitely no one-man show.
So speaking of data generation, is the data generation still ongoing for MoTrPAC?
Yeah, so there are there are many levels and many layers of data generation. Right now, there's still chemical analyses that are active, that are going on samples that have been already collected. I do believe that randomization has completed, so we're at least that far along in the human study, and all of the rat data has been collected, but we're still generating the chemical analysis, some of the human data. Malen can confirm whether or not all of the rat analyses have been completed that were planned, at least.
There are still a few that are being analyzed, but the majority. So for the rat, we've published on the adult rats, sort of young adult rats, and then we also have the aging cohort of rats. So those are being analyzed now, and we do have a few more analyses that are ongoing and assays that are being run for those. But the majority of the rat assays that are planned have been conducted already, but lots of analysis work still ongoing.
In terms of what we in the consortium sort of think of as the main human cohort, which is the sort of sedentary adults that were randomized and went through the 12 weeks of training, or non-training, as it were, for the control group. That those data are still being generated and are still forthcoming. But we do have a subset of adults that before the COVID shutdown, which affected the consortium and did many others, this group was of a couple hundred individuals, was sort of orphaned out before the shutdown. Those individuals who at least went under an acute bout of exercise, all of those data have been, of course, generated, but have also been now analyzed, and that data is under sort of the last stages of preparation for a manuscript. So we're trying to get the data out that we have to the community as quickly as we can, even if we don't have that full, the quote unquote main study data ready to go just yet.
So you both have mentioned some publications a couple of times. So I did want to sort of talk a little bit more about a couple of those. But I know that you have a publication that's sort of like taking a large snapshot across quite a bit of the rat data. So I was wondering if you could walk us through at least maybe the key physiological or molecular findings you observed in that sort of big snapshot paper first?
Sure. So yeah, we did one large study that was published last year in Nature, looking at the endurance training in these rats. So this was both male and female rats that trained for 1 to 4 or 8 weeks. So we had this sort of gradual increase in duration of training. And from a physiological perspective, we saw the expected increase in aerobic fitness, or VO2 max, and this is, for VO2 max basically assays the body's, it looks at the body's ability to utilize oxygen for energy production. And so VO2 max then is the maximal capacity for the body to utilize oxygen. And this is something that improves typically with endurance training. And it's really important, as it's one of the best predictors we have for mortality in humans. And we did see robust increases in aerobic fitness in these rats. We saw it in both the male and the female rats to a similar extent. So they all improved. We did see a difference in the effect on fat mass. So we saw that the male rats lost fat mass with training. However, the female rats stayed the same over the course of the training. We also then, in humans, we can take samples before and after, but we can't do that in the rats. So for the rats, we have a sedentary control group instead that's just not doing anything for those 8 weeks. And if we look at the females there, they actually increase in fat mass. So there is an aging effect in the female rats that remain sedentary. So they increase in fat, and this increase is prevented with training in the female rats. So prevention of increasing fat mass in the females, whereas the males instead lost fat mass. So that was a clear sex differences that we saw. But of course, we're mainly interested in the molecular aspects. And if we look at, these were 19 different tissues, so a ton of tissues, and we looked at lots of different levels of molecular regulation. So overall, when looking at all of these different time points and the training, we saw over 35,000 different molecules that changed in level or abundance with the 8 weeks of training. So it's a ton of data in there. And I think some of the key sort of take-home messages were that many of these differed between the males and the females. So there were huge sex differences in the response, and this was super interesting. We saw tissues such as like the white and brown adipose tissue, the adrenal gland, the colon. Like those are two tissues that have really not been investigated at all before with exercise training, and they responded a lot. So a lot of these unique tissues, and we also saw that a lot of the response was tissue specific. So many of the molecules that were affected, comparing different tissues, they actually responded very differently. Classically, we think about exercise, as affecting the working muscles, so the skeletal muscles and the heart, that has to work harder. Maybe some of these energy generating organs, or the liver that's releasing fatty acids. But we really saw effects across almost all of these different organs, which was super exciting.
So I'm super biased because my original background was neuroscience. So I'm curious, was there any particular neuronal differences that popped out?
Yeah, that's a great question. And we looked at 3 different brain regions in the rat. So the cortex, the hippocampus, and the hypothalamus. Looking at the global level, we did not see that much change with the training. That said, the samples were taken 48 hours after the last exercise bout. So it's really looking at these sort of more of stable levels of change rather than the transient acute changes. So it might look different with the acute exercise that we're analyzing now in the rats. And also, as with many tissues, like this MoTrPAC has looked at the global changes. It's not cell specific, so it might be that certain neurons or glial cells or specific cell types within these regions of the brain change. But globally, we saw surprisingly little actually changing in the brain.
Interesting. Okay. So I did want to back up to one other thing you mentioned about the sex differences. So you'd mentioned that there was this pretty big difference between females and males, and how for the female rats, it sort of like protected against weight gain more so than like weight loss. Is that something we also see in humans? And should I be sad as I get older?
Yeah, I guess typically endurance training is not great for fat loss in general, not speaking about men or women in particular. Uh, when we increase our level of exercise, we typically tend to compensate by eating more. So most people actually maintain a steady weight. So if you want to lose weight, you actually also have to go on a diet. I'm not aware of any clear sex differences, but that said, we're hoping to be able to answer some of those questions with the big human study when we'll actually have the power to do that. Most exercise intervention studies that have been done so far have been on smaller cohorts where, and most of them have been done only in men too. But even
Oh,
the ones that
interesting.
have included both men and women have been very small and typically analyze everyone together. Yeah, historically,
Okay.
exercise science has really focused on men.
Huh. Okay. I know that I'd read a very small number of research on sort of like men and women's bodies and potential for like fat burn being different, given hormones and sort of, wanting to hold on to fat more for potential baby making. But I don't know that I'd really seen it more on the size of like on like exercise as opposed to just like fat, gain and loss. So that's interesting to know that that is indeed the case, that a lot of the exercise is more male heavy. Okay.
In fact, Malen and I worked on a review where we took a smattering. We didn't. It wasn't a comprehensive set of analyses, but we took a lot of the big ones. And MoTrPAC like a massive outlier in terms of its size, but also in its effort to be truly both males and females as well balanced, and then of course across ages. So the goal, of course, in the human study, similar to the rats, is to start to look at some of these sex differences. The rats, because they're much more genetically similar, obviously much more genetically similar than, say, the humans, which are actually rather diverse ancestrally, which is a goal of MoTrPAC. You need a lot more of them to start to look at these sex differences, age differences, which we're in a position to sort of generate some hypotheses in the rats and then bring them over to the humans and start to take a look at them to see if we see them in the humans too.
That's super cool. You'd also mentioned you've mentioned aging a couple times now. I believe you also have a paper on some of the age effects and exercise, at least in the rats, correct?
Not yet.
Not yet.
Um,
Okay.
we're in the process of analyzing those data.
Okay. Very cool. Um,
There is, well, actually, I should add that yes, the physiology of the aged rat has been
okay.
published. So maybe that's what you're thinking
That's
about. I
definitely
just
what I must have stumbled on. Yes, thank you so much for
yeah
being more specific.
yeah,
Okay, so
and
that the
then yeah.
what is the physiology aspect involved then? Like what kinds of things are you measuring there?
So for the physiology in the rats, it's the what we talked about before, the VO2 max test for
Okay.
aerobic fitness. We're using a slightly different method in the rats compared to humans for looking at fat mass, for example. So it's NMR in the rats. And there we did see an aging effect. So as I said, the female rats did not lose fat mass with the training in the young adult rats, but they did lose fat mass in the aged rats. So there might be some aging differences there. And rats don't go into menopause in the same way as humans do. So it's slightly different there, but that's something that will be super interesting to look at, whether we see differences in the response between pre and post-menopausal women, for example, in the human study. And we did see similar increases in the response to in the aerobic fitness. And also in some, so for the phenotypes, we're also looking at some of we're looking at lean mass changes. We're looking at some more tissue specific, like in skeletal muscle, we're looking at glycogen content. So one of the known adaptations to training is we increase our glycogen content to be able to have easily accessible carbohydrates for training. And so that's also something that's included there, in addition to a few other like biochemical, looking at some enzymes that are good marker for endurance in muscle. And so a lot of that data has been published for the rats, but none of the molecular changes. So we're really excited to start looking at whether the differences in for example, then the loss in fat mass, whether that's reflected in the response in the white adipose tissue in the aged versus the adult cohort of rats. And adipose is one we have, for humans. So that would be
you
very
just
exciting.
reminded me of the carb loading that we always did right before volleyball games.
Yep,
There
carb
was a
loading is
reason
very
for that.
important.
So like see like we've talked about a couple of different publications and still ongoing work. Um, are there sort of particular pathways or biomarkers identified so far that do potentially hold promise for, some sort of clinical translation?
Yeah, I think that, that's a bigger question even than maybe we have time for, only because when you talk about the biomarkers and clinical translation, I think that inherent is that is a couple of different questions about how we want to translate MoTrPAC to clinical application, right? So if I'm a clinician at a bedside with a heart failure patient, like what do I want to use from MoTrPAC to help that individual patient? And in fact, there's a group within MoTrPAC that is specifically focused on clinical applicability, and they have a paper coming out that is exploring some of the interests that clinicians have about what they feel like they want to get out of MoTrPAC. But I think there's a couple of different key questions that we want to try to answer. So the first is like, is there a pathway that we could tap into therapeutically that would help people either respond more robustly to exercise, and also that would sort of counteract some of the effects that we see in diseases that are sort of exercise responses? So that would be, for me, cardiovascular disease is the one that comes to mind, but there are others, of course, malignancy, cognitive issues, all those, right? So in terms of those types of pathways, I think one of the areas that we're most interested in are exerkines, these molecules that are generated by, usually we think classically exercising muscle, so those would be myokines, but there are other tissues that generate signaling molecules during and a bout of exercise that basically tells the rest of the body, hey, we're exercising right now, start adapting to that exercise. And so there's lots of exerkines that have been defined out in the world that are really critical for sort of stimulating the responses globally. And MoTrPAC is sort of optimized to identify a few of those. One of the ones, obviously, the human data is unpublished yet, but there's some results that we've been talking about at conferences and such that I can talk a little bit about. So one of them is a molecule called CCN1. CCN1 has been shown to be secreted from muscle and adipose just regularly, and it's really involved in angiogenesis, in particular the extracellular matrix of the vasculature. And what we see is that we see during exercise, those levels come up in both muscle and adipose tissue during the exercise bout in humans, and that we actually then detect the levels of that molecule going up in the bloodstream. So it's being secreted and it's circulating through the system. And so the thought is that perhaps that this molecule could be driving downstream processes, not just locally in the muscle and the adipose, but maybe elsewhere. That remains to be seen. That would require follow-up experimentation, but we're interested in highlighting it as a new potential exerkine. And so that pathway, angiogenesis, is really important for adaptation to exercise, but interestingly has also been shown to be upregulated as a biomarker of heart failure severity, presumably as a compensatory mechanism. So more CCN1 is being ramped up to try to improve vasculature adaptation when people's hearts are weak. The thought is that it's also being ramped up by exercise. So the interaction between those two is a totally sort of new area. And so that would be the kinds of things that we're looking for, but they're still very early. The ability to actually translate that finding all the way to a clinical application requires many more steps, right? The question, is it druggable? Is it something that is safe to do? By the way, angiogenesis, also pretty important in malignancy transformation. So you got to be careful. Can't necessarily translate all these findings directly because there are lots of brakes on them as well. It's not just the upregulation, it's also the compensatory downregulation. So we're taking it step at a time. We don't want to oversell things. But then the other big category is, you mentioned biomarkers. We are very interested in the main study in trying to understand, are there molecules or sets of molecules that indicate or signal whether or not someone is likely to have a robust response to a training program, right? That's a really key question that would be great to bring to the bedside if a 75-year-old female comes to me and says, what's going to give me a good optimal outcome? What kind of exercise should I do? It'd be great to tell her from a molecular perspective, hey, if you tried to do an endurance training program, the data would suggest that you're less likely to have a robust response, that actually perhaps resistance exercise is more likely to give you a response. We don't have the data for that yet, but that's a separate kind of clinical applicability question that we're really, really interested in, and something that I think we will be able to answer when the training data is available in the main study.
So I'm really curious, Dan. So you're a cardiologist by training, and yet you ended up in sort of the world of bioinformatics. Like, how did you get there? Like, what was your path? And I think this is kind of useful because, I'm thinking about all of the bench biologists who see this wealth of data and go, oh my gosh, this is a lot. Like, how do I even get started? So I'm curious to hear your journey and if it might help others with sort of their journey into bioinformatics dabbling as well.
Sure. Yeah. It started really slow, and it's a gradual uptick. So when I was in medical school, I knew I was interested in cardiology. And I went to basically, I decided pretty early that heart failure was the area that I was interested in. And so I went to talk to one of the well-known sort of excellent mentors in heart failure to ask what I should do. And actually what he told me to do is to stop doing medical school for a year and do research with him instead, which was like not my plan at all. And I had to think about it for a little while. And I definitely got some advice to say, maybe you don't do that. Maybe you just be a clinician and you stay on the path. But I'm super glad that I did not listen to that advice and instead listened to Sanjeev Shah, who was my mentor in medical school, to jump off the path and do something a little bit different. Because I do think that kind of, unexpected change is really valuable in developing a career. And so when I got into that, that's when I got into just high dimensional data. So he was not working at that time with multi-omic data. We were doing something more along the lines of phenomics, which we didn't really use that word at the time, but is really what it is, which is high density phenotyping data from echocardiograms and EKGs and other things like that. Stuff we have in MoTrPAC as well, by the way. But we were really just focused on using that to apply to heart failure. And so then I thought, well, this is super cool, and I got more familiar with coding for statistical analysis. I took a couple of courses on that, got a little bit more interested in it, and then when I went to residency, that's when I became interested in like, well, okay, if a little bit of data is good, a lot of data has got to be better, right? So more high dimensional data, and that's when the multi-omics stuff started to become part of that. It was sort of a natural transition to work with that. That meant I had to learn how to do more detailed coding in R, and I learned that during my internal residency training, and it was slow because I had to do two things. But so I think that's the other thing is that it doesn't, if you don't have time for a weekly or nightly course, then make it an every other week or a monthly and do it on your own time. I think that's the only way that I was able, it was slow. It took a lot of time, but it's what allowed me to kind of get into this other area completely and completely separate from my clinical interests.
One of the things that I think is really cool about MoTrPAC is that you all provide code repositories, pipelines, pre-processed like R objects, and all sorts of really neat stuff. Um, but I was hoping to kind of end on, if for the scientists listening to this podcast, and they really want to get involved in exercise omics, or multi-omics exercise research, what advice would you give them? And Melanie, I'll start with you.
Yeah, I would start by looking at the MoTrPAC Data Hub, which is sort of our main data sharing platform that we have for MoTrPAC. And we've also started to get to collect some data and share some data from other exercise studies through there. But just go there, play around with the data. There are, tons of questions that can be asked of the data already, and we keep adding more and more as the study goes along. And what's really cool with the MoTrPAC Data Hub is that there is something for everyone. Whether you can't write a single line of code, you can still look up, your favorite gene or protein that maybe you're studying in, the neurological disease you're interested in, or maybe in obesity, or, whatever you're interested in. You can look at how that feature changes in MoTrPAC so far in the rat training endurance data in the males and females, and you can just immediately generate like a figure and see how it changes over time. And very soon you'll be able to also compare that with the human data directly. If you are interested in running your own analysis, you can choose to download data from a particular tissue, from a particular time point, from a particular ohm. Like if you're interested in the transcriptome or the proteome, metabolome, epigenome, you can look at that particular data type. You can also go and find all the raw data if you want to reprocess using your own pipelines. So there really is something for everyone. And there are also lots of really cool interactive tools. So if you're interested in comparing what happens in the liver and the brain, and like what happens to energy metabolizing pathways in the liver and the brain, there is an interactive tool where you can go in and you can directly compare what happens in the liver and the brain, and look at what pathway enrichments that are affected across both those organs. So there are lots of really, really cool tools. And if you are getting into this data, you can always reach out to us. We have a help desk email, so you can always contact us at the Bioinformatics Center. We also host monthly office hours, we call them, where
Awesome.
anyone, wherever you are in the world, you can join our monthly office hours. And we always have one member from our infrastructure team and one member from our bioinformatics team present. So you can ask whatever questions you have during that hour. So you can just join whenever. And also, if you really want to get into exercise and omics analysis, you can we're always looking for great people. So reach out and join us. MoTrPAC is going to go on for years to come. So.
Yeah, I totally agree, which is that, what's what's amazing about the Bioinformatics Center is that I'm glad that this sort of resonated with you because that's the goal. And so the fact that you said, like, you have all these different options and ways to get it, that's exactly what we're trying to do. But the idea is that there's multiple layers. Like if you want raw data and you just want to go straight into your own analysis, you live in your own world and you don't need any help, you're like, I got this. That's great. Download the data and go nuts. If you are somebody who's like, well, I'd like a little bit of environment. I love some of the stuff that you guys did in your papers. I want to use some of the same functions and tools that you built, but I'm comfortable doing it in R. Great, the R package is for you. And now you've got the data, you've got all the tools, and you've got everything at your fingertips, and you can go into it. And then exactly as Milena is describing, if you're not a coder, but you've got a mindful biology, like a lot of the people that we get to work with in MoTrPAC, you can go straight to the data hub, and there's code-free ways of investigating the data. And I will share that when we've presented this at conferences and to people, biologists and computational biologists alike, it's a little bit of an aha moment of like, so you're saying that there's essentially data in the data hub that no one has directly published on, that we could just pull right out of the data hub and use for our grants? And the answer is actually yes. It's provided by a commons license so that you can utilize that data, use it for grants, use it for papers even. We were trying to democratize this data as much as we possibly can. And I think that when you're outside looking in, there's always this assumption of like, well, the people in MoTrPAC probably already did all the cool analyses. So, I'll read their papers, but I need to go make the next data set that's going to be great. But the reality is that there is so much data in MoTrPAC that there is lots of stuff that we're just waiting for people to come and analyze and put together, and particularly to merge with their own data. There are so many different ways that could happen, and we're actively working on more and more tools for you to bring your own data and merge it with the MoTrPAC data for almost immediate insight. And so I hope people listening will leave with the impression that there's a lot yet to do in MoTrPAC. The hope of our consortium, of the NIH, and of everybody involved is that actually everybody, that there's lots of new insights that beyond what has already been published, because there's just so much.
I absolutely love that and echo that. I just finished teaching an RNA-seq workshop last week, and I pointed out that, so much of the data in public repositories for transcriptomic data, people just look at gene expression. They never look at things like splicing, exon usage, RNA modifications, and editing. All sorts of fun goodies are in there to explore and to publish on. So I just wanted to really echo Dan's point of, the data reuse and publishing, really interesting papers from public data that is open, is such a huge advantage, especially for universities that maybe, have less funding to be sort of generating their own data. This is such a wealth of resources for everyone. So thank you both so much. Do you have any other sort of parting words before we sign off for the day?
Just to say thanks for having us, and we hope to see a lot of listeners at office hours, and please email us with questions. We are excited to help people use the data.
Yeah, I completely agree. So thank you very much. It's been very nice talking to you.
Awesome. Thank you, Dan and Marlene. Have a wonderful rest of your day. You have been listening to Decoding the Data Ecosystem, a CFDE Training Center podcast. Be sure to check out our other episodes in this series. Find out more information about CFDE data and tools that may be valuable to your research by visiting our website.